Good day. I am Joshua Glenn N. Piesta, BSED2 Block 1 of Philippine College of Science and Technology. Now we are going to discuss about the difference of transcription and translation. Let us first define them. For transcription is the first step of the gene expression process. The gene is copied into an RNA piece with the aid of enzyme, RNA polymerase. This piece of RNA is called the primary transcript. It is complementary and antiparallel to the DNA sequence, which it is copied from. Transcription may produce several types of RNAs: messenger RNA, transfer RNA, ribosomal RNA, and non-coding RNA, such as microRNA. The genes encoded for proteins produce mRNAs. The mRNAs consist of untranslated regions called 5-UTR and 3-UTR for the regulation of protein synthesis. Other RNA types are considered to help the synthesis, regulations, and processing of proteins. For translation is the second step in the process of gene expression. The mRNA is produced by transcription and translated into proteins in the cytoplasm by ribosomes. During translation, mRNA is decoded by ribosomes in order to produce an amino acid chain or a polypeptide chain. Complementary tRNA anticodon sequences carrying a specific amino acid bind to the mRNA. This type of tRNAs is called amino-acyl tRNAs. The binding is facilitated by ribosomes. The amino acid carried by tRNA from polypeptide chain by peptide bond formation between two amino acids. This amino acid chain undergoes post-translational modification and then folds into 3D structure in order to become an active protein. First difference was transcription is the first step of the gene expression process, while the translation is the second step of the gene expression process. Next is for transcription, synthesizes RNA copies of the genetic construction written in the genome, while translation synthesis protein from RNA which are copied from genes. Next is transcription's template is the genes in the genome, while the translation's template is the mRNA. Next is transcription occurs in nucleus, while translation occurs in cytoplasm. Next is transcription's RNA polymerase are the enzymes, while the translation ribosomes are the enzymes. Next is transcription produces several functional forms of RNA, while translation produces protein. Next is transcription localized in prokaryote cytoplasm and eukaryotes nucleus, while translation localized in prokaryote cytoplasm and eukaryotes ribosome on the endoplasmic reticulum. Lastly, for their difference, transcription is inhibited by rifampicin and 8-hydroxyquinoline, while translation is inhibited by tetracycline, chloramfenicol, streptomycin, and cyclohexamide.